All right, guys, I think I'm recording, although who knows with my iPad these days. And so I'm going to try to start a timer here and so that I don't go too long. And I'm going to go really fast. But you should have opened up the notes. I think I called them Snow Day notes. Unit 4 cell division is what we're starting. And I'm going to talk fast, knowing that you can pause if you need to write things or review things. But I'm just going to kind of talk fast. A lot of this is an overview of stuff you had in Honors Bio or stuff that's in the notes in more detail. And I'm really counting on you guys to be AP here and kind of dig in and read the notes and do the things yourself. So these are the things I'm going to talk about. I have bulleted here. You guys already did notes last night on sexual versus asexual reproduction. I just want to go over that. Diploid versus haploid, that's review. Chromatin versus chromosomes, an overview of mitosis and meiosis. And then what we're doing now in this chapter, this is chapter 11. Chapter 11 is this is about mitosis, okay? And then chapter 12 is going to be meiosis, and we'll do that later when we come back. So I already started some notes here for you to copy down if you need to pause so you can write some stuff down. And just sort of my overview here of asexual and sexual reproduction. And so kind of the overall thing that I think is, the clarifier is how many parents there are. So asexual reproduction is one parent reproducing, whereas sexual reproduction we'll see is two parent, although we're going to see not always. There's some examples where it's not always two parent. The real definition of sexual reproduction, this is sort of here, the real definition is that if it requires meiosis to make haploid gametes, oh wait, that's not the real definition, sorry, the real definition is going to go down here. Okay. So the real definition is if two haploid gametes have to fertilize, my pen's not working. Fertilization of two haploid gametes. So if there's a fertilization event, which means these are kind of some words I'm hoping that you kind of know, two haploid gametes. If you remember, haploid is a cell that has half the chromosome number, and gametes are what we call the sex cells also. Gametes, the sex cells, that's like, for example, that would be sperm and egg, okay, in humans. So the real definition of sexual is if there are two haploid sex cells or gametes that have to fertilize in order for there to be reproduction, we call it sexual, okay? Now, usually that's from two different people, two parents, a male and a female, kind of like with humans. But what we're going to see is not always. I'll give you an example of not always here. Let me kind of draw up here. There's some worms, roundworms, that, here's my worm, that are hermaphroditic, meaning they produce sperm, which are haploid. We use the little letter N or any letter N to mean haploid. So they can produce sperm, and they can also produce eggs, which are also haploid, just like we produce sperm and egg, but we produce them in a male and a female. Like a worm is hermaphroditic, and it can produce both. It might have the sperm production at one end, egg production at the other end, and it can fertilize itself. Okay, that's one parent. That's one worm, if you're thinking of it that way. But we still call this sexual reproduction because there's a fertilization of two haploid gametes that have to occur. Okay, so that's why I mean two parent, not always. But in general, that's kind of the way to think of them. A lot of times, so roundworms, like some of these species, they can fertilize themselves if they're in a situation where they don't encounter another organism, or two worms can kind of line up next to each other, and this one fertilizes that one, and that one fertilizes that one, which is, still two parent, but they don't have male females. They're hermaphroditic. So there's a bunch of gray areas in here. I'm just kind of giving you kind of the overview of sexual versus asexual. And it has to do with the types of cell division that we're learning. We're learning about mitosis and meiosis, and those are involved in these two types of reproduction. So asexual reproduction, like I said, is one parent. Sexual reproduction is two parent. And kind of our example, let me see if I can draw a picture here. I didn't leave myself much room. A sperm and an egg. So here's our example. Let's say humans, our chromosome number is 46. And the way we write that is 2n equals 46, because 2n stands for diploid, okay, which we'll get to in another slide here. But we have 46 chromosomes, which means that when we reproduce, we have an egg that's haploid that has 23. We have a sperm that's haploid that has 23. So this little n means haploid, means it has half. Okay, so those are my haploid, those are my gametes or my sex cells. They kind of mean the same thing. And they obviously have to come together and fertilize. We have a fertilization event. And then we get this diploid, I'm going to call that's 2n, zygote. So that's the first cell after sperm fertilizes egg. And then from there, I'll kind of draw it over here. Then we would have mitosis, which those cells would then divide into identical cells. These would all have 46. This would be an embryo, which is, starting the multicellular. I'm just going to 46 in all of these, okay? So, that's kind of how we reproduce. We all, I think, understand that. The key here is that there's a fertilization event. Okay, so we have a fertilization. Fertilization is when you go from haploid to diploid. So you have two haploid things that combine and you get diploid, which is 2n. Okay, and then once that zygote forms, then we need mitosis to just divide into... Mitosis is creating identical cells with the identical chromosome number. And so I set up here... it requires, let me pick a different highlighter color here. So sexual reproduction requires meiosis to actually make the sperm, the gametes, and the sperm and the egg. And so we're going to see that's in chapter 12, and I'm going to give you an overview tonight. It's going to be cutting the chromosome number from 2n to n. So meiosis is the type of cell division that specifically is for making haploid sperm and egg. That's it. So in your bodies, the only place you do meiosis is the ovaries to make eggs and the testes to make sperm. The rest of your body doesn't do meiosis, okay? So sexual reproduction involves meiosis to make these haploid gametes, okay, that we're seeing here. Those are made by meiosis. But then it also requires mitosis to grow, which we kind of saw here. Once you have the fertilization event and you have that one cell, it has to multiply into a multicellular organism by mitosis. Over here in asexual reproduction, we only have mitosis. There's no meiosis. And so they reproduce just by doing mitosis, which is the identical cell division only. And then these are all the examples that are in the notes of asexual reproduction. I had you read about them last night. I'm going to quickly fly through them, but I don't think they need much explanation. You guys can read them on your own. But these are all examples of a living thing that can reproduce all by itself. Doesn't need a mate, a fertilization, anything. Okay. So the other thing I want to go back up and mention that's kind of important is that in asexual reproduction, we get identical offspring. Okay. Because it's just one thing reproducing an identical clone of itself. Whereas in sexual reproduction, this is kind of a key, you get variety in the offspring, right? Think about humans. We don't all look the same. And that's actually better for the species. And we're going to see that's better for the species in terms of when we talk about evolution and being able to adapt to a changing environment. Variation is key. Because if our environment changes, some of us might die, but some of us might have traits that will survive. And so as a species, it's better to have variety. Like this is kind of a key thing to understand. So sexual reproduction, if we look at it in terms of time, asexual reproduction came first, in terms of life. And sexual reproduction kind of came about later. It's kind of more advanced, more complicated. And it's better for this reason, because you get variety in the offspring, and that's better. And when we get into evolution, we'll get more into that. So I'm going to skip through down here and just some of the slides that are in my... These were my PowerPoint that I posted already, and it's kind of hard to read this anyway. But I want you guys to make sure you go through these, and I'll point out a few here. Like it talks about, asexual versus sexual reproduction, one parent, two parent, single parent, all that stuff about being genetically identical, genetically unique. This talks about kind of the advantages and the disadvantages of each. And so asexual reproduction, there are some advantages to it. It's more time efficient, and you don't have to search for a mate, and there's less energy involved. So they don't have to produce gametes and hormones and have mating rituals and things like that. And it's an advantage if you're a living thing that's living somewhere where there's no chance, where you're going to, where is it? There's very little chance. That's not what I wanted to say. It doesn't say. Where does it say it? Or you have no chance of running into another person, okay? So let's say you're a starfish out in the middle of the ocean, okay? Maybe you're never going to encounter another starfish because you're very spread out. And so it makes sense that you would evolve to be able to reproduce all by yourself. Otherwise, starfish would have gone extinct, okay? So there are some advantages to asexual. You can reproduce all by yourself. You don't have to search for mates. You don't have to make gametes. It requires less energy. It's a lot easier, quicker. The only disadvantage is you have no variety, okay? In sexual reproduction, we'll notice that most of the advantages have to do with variation, okay? Variation is the key to evolution, and species are more likely to adapt and change to their environment. So that's the main thing. So make sure you kind of read through all of these. These just give you examples of what kind of living things do sexual versus asexual. I'm not going to go through that. It's all in the notes. But I just wanted to kind of show you some pictures here. I don't know if you remember, like from freshman year, I think I showed videos of like a hydra. The hydra is a tiny little, this is actually an animal, and it's like a jellyfish. It's in the same family as the jellyfish, and it's like an upside down jellyfish, and it has these stinging tentacles, but it kind of adheres to the bottom of the lake or whatever. And these are really small. They're probably like not even a centimeter tall. And they're green oftentimes, so people think it's a plant, but it's not. It's an animal, and it stings things and eats it. But what you can see here is the way that it reproduces is by budding, and so you just have mitosis going on here. Mitosis is just identical cell division, and you see that it's just growing a new hydra out of the side. It'd be like if I just grew a new Mrs. Flannery out of the side of my body, and then it's like a little, and then all of a sudden it comes off, and then I have a little Mrs. Flannery. And so you can see, so here's the, the little clone baby. It's going to be an identical in terms of DNA and genetics because it just divided by mitosis, which is making identical cells with identical chromosomes. So that's budding. Fragmentation, this is a little planaria worm, where basically you can cut it. But basically, if you cut it in half, you're going to have the head grows a new tail by mitosis, and the tail can grow a new head by mitosis, and then you end up with two. So it reproduced one worm into two worms just by cutting it in half and growing new cells by mitosis. So obviously, complicated organisms like ourselves, more highly evolved, this wouldn't work. It'd be too complicated. A lot of plants reproduce asexually by vegetative propagation. Again, you just have mitosis kind of here. There's a little stem that's a runner that grows off, and then it grows new roots and eventually you have a new plant. It's a clone. Okay. So strawberry plants are known to do that. Here again, it talks about hydra and budding. Fragmentation, starfish do that too. Just parthenogenesis. I'll let you guys just kind of study through that. I don't want to talk too much more about it. Asexual, binary fission. Remember when we did, that's how when a bacteria cell divides, we call it binary fission. An amoeba is also a unicellular, it's a protist, it's not a bacteria, but it's a unicellular organism that just divides in half to reproduce. And so that's called binary fission. And here I wanted to give you another example. So let's talk about sexual reproduction. And I wrote up here, not always two parents. And here's another example. So here's an example kind of in flowering plants. And if you've ever looked in the middle of a flower, you have all these little stemmy things in the middle. And there's one thing kind of at the bottom. There's kind of one stem that's kind of in the center, and there's eggs in there. So those are haploid gametes. There's little eggs down in there. Okay? And then there's usually, a bunch of these little things coming out of the sides, and that's where pollen is. And so this produces pollen. Pollen is like sperm. It's just like flower sperm. And so pollen can land in that same flower and fertilize. It can self-fertilize. And so that's still sexual reproduction, because there's a fertilization of two haploid gametes, even though it's only one parent. Okay? I mean, plants can also cross-fertilize, but just because it's one parent doesn't mean it's asexual if there's a fertilization. I want you to familiarize yourself over here with this kind of human life cycle. I think you all probably know it. I mean, it's us, basically. But looking at the N and the 2N, remember, N is haploid, so that's half. So you have your egg cell that's half and the sperms that are half, and those are created by meiosis of the males and the females. And then when you combine those together in fertilization, you get your first diploid zygote, which has got all the chromosomes, 2N, 46 if it's human. And then that goes through mitosis to divide into a multicellular organism. Okay, I love how I have naked people here in the notes. And then, we start over. So in our life cycle, part of our life cycle is diploid and part of it's haploid, and that's what you can see by this kind of cutting here, the blue and the yellow. So the yellow is the haploid part of our life cycle, which is just the sperm and the egg. And then the rest of our life cycle is all diploid with 46 chromosomes. Okay, so that's all sexual reproduction. And then I mentioned that in sexual reproduction, the sperm are made in the testes. Okay, so the only place in a human body that's doing meiosis, so this is where meiosis is going on, and it's producing sperm in the testes. Okay, and in a female, the only place that meiosis is going on is in the ovaries, and it's making eggs. Because remember, meiosis is a specific cell division that just makes sperm and egg. All right, so I'll let you guys kind of read through that. Diploid versus haploid, I've been sort of talking about it, so I don't think, and it was in your homework last night, so I just want to kind of go over it, some of the terminology here. So each species has a characteristic chromosome number, all right? And we call that the diploid number. So humans have 46. That's like our diploid number, meaning that's how many we have for a normal human. Like chimpanzees have 48, chickens have 78. Don't memorize that, but obviously notice the number of chromosomes doesn't necessarily mean more highly evolved. Like a chicken has more chromosomes than us. That doesn't mean they're more complicated. It just means that their DNA was divided up into more chromosomes. It doesn't mean anything. So if this is the diploid number and I asked you questions like, how many chromosomes would be in the sperm, you just cut it in half. If I'm asking for the sperm or the egg of any of these things, or the sex cells or the gametes, you just cut that in half. So diploid, we use 2N is kind of the symbol for it. And that's, it's 2N because it's going to have two of each chromosome. I'm going to talk about homologous pairs here and draw a picture down here in a minute about how we have pairs, but you can also see them over here. This is a picture of a human cell and a diploid cell, 2N, that has here's all 46 chromosomes. laid out in something we call a karyotype. They're not like that in the cell. But you can see that we have 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s, 2 chromosome 4s. And the reason we have 2 of each is because we get one is maternal and one is paternal. You get one from your mom and one from your dad in each pair because we're sexually reproducing. Okay, so notice that the haploid cells, so this would be sperm and egg. If this was a sperm or an egg and I was looking at their chromosomes, notice they're haploid, they have half, and they only have one of each chromosome type. They have no pairs, okay? Diploid cells have, we call these pairs homologous pairs. So that would be a homologous pair, okay? And so when we talk about humans' chromosomes and homologous pairs, if I say that our chromosome number is 46, we would say that we have 23 pairs. Okay, and we'll go into this later, but the way the chromosomes are numbered, largest, and then the sex chromosomes are the last pair. We'll get into that later when we do genetics. Right now, I just want you to understand that a diploid cell has these homologous pairs, has all the chromosomes. So for a human, that would be 2n equals 46 is how we would say it. Somatic cell is another word for, kind of a fancy word for a body, like cell. Basically, any cell that's not sperm or egg. That's the definition of a somatic cell. Any cell that's not a gamete, sperm or egg. And I think I defined them down here. So a somatic cell is a body cell that's not a sex cell. So it's another word. If I'm talking about your somatic cells in your body, that's pretty much all your cells, except for the sperm or the egg, depending on who you are. Okay. So it's a lot of just terminology here to get used to. Haploid, then, we said we use the letter N. It can be a capital N or a little n, doesn't really matter. It has, just remember, haploid and half both start with H. So there's no homologous pairs because it only has one of each. And the only cells that are ever going to be haploid are the sex cells or the gametes of species that reproduce sexually. Okay, asexually, if you reproduce asexually, asexual species, they don't need to ever have half. They don't need haploid cells because they're never combining a sperm and an egg or anything like that together. And so the only reason, if we go back to this picture here, the only reason we need to have cells with half is because the way we reproduce is by combining these two cells. If the eggs had 46 and the sperm had 46, then the zygote would have 92, and so would the embryo, and then every generation the chromosome number would double. That just wouldn't work, all right? So only species that reproduce sexually, where there's a fertilization of two cells, that's the only time you would ever need haploid half cells. So if we're talking about asexual reproduction, there's no, they don't need haploid cells. Okay. All right, so that's diploid, haploid. I want to just draw you a picture of homologous chromosomes and talk just briefly about it. Let's talk, this is going to come up more later when we do genetics, but so a homologous, that's an S, homologous chromosomes, or what we refer to as a pair of chromosomes. Okay, so let's say I'm looking at, this pair. So let's say this is chromosome 1, and this is your other chromosome 1, right? This is a homologous pair. I'm just calling them, we number them like 1 through 23 or whatever. And let's say this is your maternal chromosome 1, you got that one from your mom, and this is your paternal chromosome 1, you got that one from your dad. Okay. Now, homologous chromosomes, notice they're the same size and everything like that, and they're going to have the same genes on them, the same DNA genes. They'll have the same genes. For example, if eye color is on chromosome 1, which I don't think it is in humans, but just say it is. So they're going to have the same genes, like eye color, but not necessarily the same alleles. And I don't know if you remember the term alleles. So alleles would be like, you know, big B, little b. Like when we talked, do you remember when we did genetics and like big B stood for brown eyes and like little b stood for blue eyes. Can't spell eyes. These are what we referred to as, you know, the alleles. Alleles are defined as different forms of a gene. So, for example, like up in this chromosome pair here, let me switch colors. Let's say your mom gave you the eye color gene, let's say is right here on the chromosome. So let's say your mom gave you a big B allele, basically a gene for brown eyes, and your dad gave you a little b allele, a gene for little eyes. And so you'll notice like both chromosomes have all the same genes, like eye color, hair, whatever, if it's eye color and hair color and tongue rolling and whatever, they both have a gene for eye color in the same place, but your mom's and your dad's form of that may be different. Those are alleles, okay? So that's a homologous chromosome. And then we're going to see, sometimes we draw chromosomes like that, and this is where it gets confusing. Technically, that never exists. Well, yeah, it does. But a lot of times we draw them like after it replicates. Okay, so let's say that's your mom's. It's going to replicate, make a copy of itself. And so a lot of times we see them chromosomes drawn like this as X's. And so if your mom's original chromosome had a big B brown eye gene, then that means this has a brown eye gene, because these are, remember, what we call sister chromatids, and they're identical. Because one was a copy of the other. When that's going to happen during S phase. And your dad, you know, if you started out with a little b, a blue-eyed gene here, then when it made a copy of itself, this is going to be, you know, these are also sister chromatids, identical. So technically, and I'll get into this a little later, when we draw, these are two replicated chromosomes, okay? So this picture, a lot of times people would say, oh, that's two chromosomes. I see two X's. But this is what we call two replicated chromosomes. Meaning it's really 4 chromosomes, because this was 2 chromosomes. This was just 2 normal chromosomes. And then each one made a copy of itself. And now kind of there's 4 chromosomes here, but we never say that. They say there's 2 replicated, but in your brain, I want you to just think that's kind of like there's kind of 4 there, okay? And then in mitosis, we're going to see these are going to line up. And remember, this is what we call the centromere here, kind of the middle, which holds the 2 sister chromatids together. And they're going to line up and then split in half in anaphase. So those are homologous chromosomes and homologous pairs. And you see those only in diploid cells. We're going to see them when we go through mitosis. So let's look at the cell cycle really quick. Let me check my time. I don't want to talk too long. Okay, so the cell cycle, you guys kind of watched a little bit about this. Actually, I want to skip and let me do chromatin versus chromosomes first. We already talked about this when we did cells, but let's kind of review it. So if you remember, this is all chromatin and chromosomes. These are all DNA. This is 2 different ways your DNA can exist in a cell. And if you remember, chromatin is when it's kind of, it's like an unwound sweater, and then chromosomes are when we kind of supercoil and wind up the chromatin DNA, like you're knitting a sweater that looks like an X, okay? So I like to think of it like a chromosome is like a sweater. It's a string. It's made of these strings. If I pull like a string, it's going to unwind into a big, long string. That's chromatin. And remember, our DNA in the nucleus, sometimes it's unwound as chromatin, and sometimes we scrunch it up into chromosomes. And the only time that we ever scrunch it up into chromosomes, and you can see all of this here. So this is a chromosome, which means it's chromatin or DNA that is supercoiled and condensed and coiled and coiled and coiled and coiled. And when it's like that, the genes are not exposed. So like any DNA that we wanted to read what was on it, we couldn't get in there and read it. So the only time you're ever going to see this is when the cell is dividing. That's the only time we ever take our DNA and scrunch it up into chromosomes, because it's going to be easier to line it up and divide it and all that stuff if it's in these little X's than if it's all like spaghetti. Okay, so it makes it easier for the cell to divide it up. So the only time chromosomes are going to form is in this process we're doing now, mitosis. Chromatin over here, we call it sometimes uncondensed. It's like an unwound sweater. But the genes are exposed, meaning like in a normal, this is where most cells function. Most cells that are just doing their job, their DNA is unwound like this, where you can get in and read the DNA that's in there, the genes. Okay. So I was going to do a quick little, let's do chromatin. Sorry. I want to do a different color, and I want to do, okay. So chromatin versus chromosomes. And this is going to help you understand what's going on in mitosis too, when we talk about the chromatin condensing into chromosomes, etc. So I like to think of chromatin as think of it like stringy DNA, or a better way of saying it's uncondensed. Uncondensed or unwound. Those are kind of the ways we kind of refer to it. Okay, and this is what, let me put a little star. Most cells, this is how the DNA is, because most cells are not dividing. Okay, they're just a normal cell. They're doing their job, and so kind of what it looks like. Let me go a little bit thinner here. Okay, so let's say if this is a cell, and that's the nucleus. Actually, I'm going to switch colors here. And let's say that we're dealing with, I don't want to draw 46 chromosomes. Let's not do a human cell. Let's say we're dealing with something where the chromosome number is 4, okay? So the diploid number is 4, which remember would mean it has 2 pairs, right? 2 homologous pairs of chromosomes. So what the DNA would look like, remember chromatin, we said this looks like spaghetti. That's chromatin. You can't see the little, how many there are. It's all uncondensed. It's stringy. That's what it looks like. And we're going to see that during interphase, okay, of the cell cycle. If I were to look even close, let's see what this looks like. Okay, I said there's 4, the chromosome number's 4, but when it says chromatin, let's say that's 1, and I'm going to draw where the centromere would be. Let's say that's one piece of one chromosome. It's going to be a chromosome, but right now it's 1 chromatin. And let's say that's the other. So let's say that's chromosome 1 and chromosome 1. So that's a chromosome, that's a homologous pair. And then the other pair, maybe it's a smaller one. Okay, but the pair, so let's say this is going to be chromosome 2, and that's going to be the other chromosome 2. Okay, but right now they're uncondensed as chromatin. And this could be maternal, paternal, and this is the maternal and the paternal of the homologous pair. Okay, so remember, so this is, these are the pairs, what we call homologous pairs. Okay, so it has 2 homologous pairs, but they're existing in the nucleus right now as unwound chromatin. Okay, so what's going to happen then, let's say this is what we're going to call G1 of interphase, which we'll go into in a second here. And if you remember, I think you guys watched this last night, the other part of interphase is called the S phase. So remember, interphase is broken into G1S and G2. And the S phase is where the DNA makes copies of itself, or the DNA replicates. Okay, so this is still part of interphase. And so what you're going to see then after that, let me make this arrow a little shorter so I have room to draw. So the cell is still, let's say here's my cell, here's my nucleus, it's still, my 4 chromatins are in there, okay? But once it makes a copy of itself, I'm going to run out of room here. I'm going to do the copy in red. So in S phase, when the DNA replicates and it makes a copy, and so that one's going to make a copy of itself. It's all attached at that centromere, and I'm going to draw the copy in red. Okay, and see, it kind of looks like an X. I didn't leave a lot of room here, but these are supposed to look kind of like stringy X's. All right, so remember, these are the sister chromatids, the blue and the red, and they're identical. The red is a copy. This is a copy of the blue DNA. Those are your DNA. Okay, so again, remember, like when we were doing, if we were putting if we had actual alleles on here, if there was a big B there, and then there's a big B brown-eyed there. There's a little B there, there's a little B. Anyway, so the sister chromatids are identical. So that's still stringy. So in the nucleus, though, my DNA still is stringy, but now it's kind of like double, right? I have double copies. So this is still interphase, and this is all still chromatin. Okay? Then what's going to happen, then we're going to get chromosomes. Okay, and this is going to happen during prophase. And that's when it's going to condense down into, let me go back to my blue and my red. So each chromosome is going to kind of supercoil and wind up. So I'm going to end up with, let me draw 4 condensed chromosomes. Let's see if I left myself enough room here. Do those look like X's? Not really, sorry. Those should look X-y. Okay. And then the other ones, whatever, were a little smaller. So the DNA, the chromatin, just supercoils and condenses into chromosome shapes that look like X's. Okay. So these are chromosomes now, all right? So if we're dividing this picture, right, chromosomes are, not stringy DNA. We call them condensed, DNA, or it's supercoils. And this is only during cell division or mitosis that it does this. And specifically, it does it in prophase. Okay. All right. So we have chromatin, single copies of each chromosome as chromatin. So we started out with 4. Each one made a copy. So we have 4 replicated chromosomes here, but it's really 8. And then we're going to condense them down into smaller little shapes. These are much easier than these little chromosomes. We can line them up in the cell. We're going to get rid of our nucleus because it's in the way. Oh, so I didn't draw. So what the cell would look like. Oh, this will take too long to draw. But I think we get the idea. We have our, we have our chromosomes now in the nucleus. All right. So eventually what we're going to do, so that's prophase, is we're going to get rid of, whoops, we're not going to get, well, actually I was going to say that. We're going to get rid of the nucleus, but I don't want to show that yet. We still have a nucleus, but eventually what we're going to see is that we're going to get rid of our nucleus because it's in the way, and then we're going to line all these chromosomes up in metaphase down the middle, or the equator, split them in half. So each one, the red and the blue copy, are going to go to each new cell in anaphase, and then it'll form a new nucleus and split in half. And that's essentially mitosis. And so the phases of mitosis here, I'm not even going to go through them that much, but I want you should remember those and what's happening at each phase. But you can see here in interphase, here's my DNA as chromatin. It makes a copy of itself while it's in this phase. Then in prophase, now my chromosomes are forming. My nuclear membrane is disappearing. My spindle fibers are forming. Okay, so the spindle fibers, remember, are microtubules that are going to form between the centrioles. And then eventually the spindle fibers attach to the centromeres and kind of go to the poles, and they're eventually then going to pull those 2 identical sister chromatids apart. Now here they have them as the same color. Pretend this one's blue and this one's red in my picture. Anaphase, just remember anaphase apart, metaphase middle. We call this the metaphase plate or the equator. And then in telophase, everything's just going back to normal. So the chromosomes are going to uncondense back into chromatin. We're going to form new membranes around it. We get rid of the spindle fibers and we do our cytokinesis. We split in half. So that hopefully sounds a little familiar. And just going back then to this picture of the cell cycle, it's the same thing we're just looking at, but sort of we see this pie chart a lot. That's why we call it the cell cycle, because it goes over and over. So if he gave you, he told you that to memorize it as kind of IPMAT, IPMAT, and we did this freshman year. Interphase, prophase, metaphase, anaphase, telophase. Just remember IPMAT. And remember interphase is broken into G1, S, and G2. That's interphase, prophase, metaphase. I don't feel like writing it. And telophase. Okay. And then we have cytokinesis. Okay, which I kind of drew as sort of under telophase. Sometimes it's considered like its own phase, like after telophase you have cytokinesis. And cytokinesis, remember, just means the division of the cytoplasm. That's what the word actually means. And when the cell finally just splits in two at the very end, we call that cytokinesis. Sometimes we consider this like part of telophase. And sometimes they refer to it as like its own phase. They're both kind of correct. They make it confusing. Prophase, metaphase, anaphase, and telophase. This is technically mitosis. So what we call mitosis proper. And remember, mitosis means division of the nucleus. So this is like the DNA condensing and lining up and dividing. Okay. Now, what's confusing is we sometimes call this whole thing mitosis. So, you know, like sometimes we would refer to, I would make the title up here, mitosis. Like this whole thing is referred to as like interphase is sometimes considered, you know, I say, what are the phases of mitosis? Interphase, prophase, metaphase. But mitosis technically is just prophase, metaphase, anaphase, and telophase. Technically, mitosis proper. And then this is a cycle, right? It's just going to start over again. And so here it is kind of drawn more in a cycle. And this pie chart is key because it gives you the timing. And so you can see here, interphase, which is this blue wheel. Most of the time that what a cell is in is interphase. And then the pie, the little yellow slice is the actual dividing. That's a very small time that the cell is in that. And of course that would vary with different cells. And something I wanted to mention, I think he might have talked about a little bit in the video you watched. So in G1, you know, we have it grows. Here's S phase where our DNA replicates. So I drew, that's where the blue chromosome turns into a blue-red chromosome, but it's still chromatin. In G2, it makes more organelles. It's kind of getting ready. It's also making the enzymes needed for mitosis. So kind of like the, let me draw like a line here, sort of like, you know, most cells are right here in something, most cells are in what we call the G0 arrest, which means they stop. So most cells get to G1 and they go into G0 arrest where they just stop and do their job. Most of your cells right now that's where they are in the cell cycle. They're basically in G1 but stopped. And they're not dividing. They're not doing mitosis. They're just doing their job. And that's where most cells are. Then at some point, let's say this cell gets a signal. It gets a signal to divide. Like maybe you cut yourself. Okay, maybe this is a skin cell. And so it's in G0 rest here. In G1, it's doing its job. It's got chromatin, etc. And then now it's going to divide. So the first thing it's going to do at the end of G1 is it's going to start to grow. It's preparing for cell division now from this point on. Then it's going to say, well, I better have double the copies of my DNA if I'm going to divide. And then if I'm going to divide in G2, I need to make more organelles. And I need to get all my enzymes that are going to be needed to run this whole process. So it's kind of preparing for division here. And so these take a lot of time. And then this little slice of the pie here, this is really labeled kind of bad. There's actually, I don't know if you can tell, but the yellow slice there's sort of two different yellows. And I don't just cross that out because I know it's kind of incorrect. But this gray arrow here, this is mitosis proper. Right? Which is your prophase. Ignore pro-metaphase. We're not going to, it's like in between. We got prophase, metaphase, anaphase, telophase. So all of that is this little slice of yellow. And then this line really should say cytokinesis. This other little yellow slice. So notice that this part is a very small slice of time. And you need to be able to recognize cells in each phase. These cartoon ones are easy. Kind of study, these cartoon. You should be able to recognize a cell in each phase. I had another document, I think he had you practice with some real cells last night, but I wanted to kind of show you because this weekend or this over break, I'm going to have you do this. These are plant cells, and so you can see the squares are the cells, and the circles that you see are the nucleus, okay? So the circles are not the cells. The squares are the cells. And I want to show you, letter C up here. This is interphase. And so the square, those are 3 cells. And the way you can tell interphase is because you sort of have a perfect circular nucleus. You have a perfect circle for a nucleus, because the membrane hasn't started to break down, and the nucleolus is still visible. So you can see in these here, this one has 2 nucleoli, it looks like eyes, and that you have this perfect nucleus. And when the DNA is chromatin, it stains kind of like a lighter, it looks like you colored in the nucleus with a light colored pencil. Okay? If you can see dark, the nucleolus is still visible, that means you have chromatin, which stains lighter. That's interphase. So you'll notice most of these are in interphase because that's the longest phase. Prophase is the one that they get mixed up with. So the interphase is there. And then let me show you, down here we have, so here's one that's in prophase. Here's another one that's in prophase. So you can see here, what you see is, first of all, the nucleus is not a perfect circle, because the nuclear membrane is starting to disintegrate. And so it kind of starts to spread out. And you also see the chromosomes are forming. They stain darker. So they look if you see dark kind of wormy things in the nucleus, those are the chromosomes condensing, and they stain darker. And so you'll end up with sort of empty white space in there. And that's how you can tell it's prophase. Okay, so this one's interphase. This one's prophase. The other ones are pretty easy. Most people can tell, that one is anaphase, because you see they're moving apart. Here, as soon as there's space, that one's also anaphase. This one is the other one that's this is telophase, where you can't really tell, but there's sort of a shading and there's a cell plate down there. Cell plate is how it divides in half by cytokinesis, and you can see that the chromosomes have reached both poles. So these pulled apart here, and then they are like this. They're starting to kind of form a nucleus again. So telophase is kind of hard. This one's metaphase, where they're lined up in the middle. This one's metaphase. D, this would be prophase. I just want you to get used to this. I don't know what FF. I'm going to say pretty early prophase. It looks like chromosomes are forming. This one's anaphase. This one's metaphase. This one's interphase, because you can see the nucleus there. So, and this one's ana. So you're going to have to do this on the homework over the break. So I just wanted to practice that. And then the last thing I wanted to do, if you go back to these notes, is the 2 types of cell division. And again, we're going to do meiosis later after break, but I just wanted you to see here. So looking kind of an overview. So mitosis here, it starts with a cell that's 2N, that's diploid. So if we're talking about a human, that's a cell that would have 46. And then after S phase, when it copies of the DNA. Remember I said it kind of has 92 chromosomes, but it really only has 46 like X's. It has 46 X's, but that's kind of like 92. So we call that temporarily 4N after S phase. And then it divides by prophase, metaphase, and then you get two identical. These are what we call the daughter cells. They're identical. They're diploid. There's only two of them. So these cells in the cell you started with, everybody's identical. The whole point of mitosis is to start with a cell with a chromosome number and make two identical copies of it. And that's the whole point. And so we use that for whenever we grow, we repair tissue. That's how asexual reproduction reproduces. So, and then we'll get into meiosis later, but what you can just kind of giving you an overview here. We start with a diploid cell, but then we end up with, down here, you notice it's going to go through the phase. It goes through IPMAT, the phases, and then it's going to go through PMAT again. So it goes through the phases twice. There's kind of two divisions, and so you end up with 4 haploid cells, but they're not identical. And this confuses people. Like if this was human, and you started with 46, and you might think, well, all 4 of those sperm have 23 chromosomes. How come we're saying they're not identical? Well, they all have 23, but they have a different combination of 23. And we're going to see it's like if I had a deck of cards and I split it in half and I gave you half. And then I shuffled it and I gave you half. And I shuffled it and I gave you half. I'm giving you the same number each time, but you're getting different cards. So we're going to see these are actually, they have the same haploid number, but we'll see the DNA and the chromosomes are very different, and that's why we said we get that variety in sexual reproduction. So I know that was a lot, and I know that was fast, but it's a mostly review. All of this is in the notes, and you're going to review it again. So you guys are going to upload these notes so I can see that you took notes, and I have the review worksheet to do, which is just a couple of quick review questions where you're just quickly answering, seeing if you understood the difference between all these things I talked about, and you're going to upload that as well. Okay?